All right, I'll invite everyone to find their seats, and I'll page my session moderators to figure out where they are. You want to stick that back up here? Perfect. Brett, Annie, Sho.
Hi everyone, welcome back. Okay.
How long do we have for this?
I think, Stephanie, how long do we have for this? 15 minutes. Okay, I'm assuming you'll flag us down because I don't have a phone or any timekeeping mechanism. So I think the goal of this little thing is really to give everyone a summary who wasn't able to be in the other two sessions other than the one that they're in about sort of some of the themes and key takeaways from the more in-depth discussions on each of the aims. So I guess we'll just go in numerical order because that probably makes the most sense just in terms of project progression. So for aim 1, we had a lot of discussion in our room about sort of the characteristics of the people you need to be involved in this kind of effort. So in terms of the patient scientists, what are the characteristics, so for future groups or if other people are interested in embarking on this type of work, what are the types of qualities you want to see in patient scientists, and also some of the lessons that we learned, because originally I don't think we actually called our folks patient scientists, and then it actually happened, we used the term and it sort of changed the level of engagement. So I think that's something as a lesson learned and something to do up front is to make sure that you do that and you sort of set the expectations and the understanding that everyone's has a voice at the table and they're there because they have their expertise, and i.e. patients have the credentials really in their disease, and that is no different or does not make them any different than the PhD or the MD or whoever else is in the room, and that you want to make sure you're sort of encouraging that dialogue and encouraging people. You know, we were very lucky, Margaret and Anna were not shy. They made sure their voices were heard, but in other cases, maybe there are patients who may not feel that up front, so you want to try to ensure that you're creating that environment to have that space and that people are all feeling heard, whether they're the patient scientist, the clinician, the reviewer, whoever's a part of your team. And another feature we realized too, not just for the patient scientists, but for our clinicians, one of the things that Katrina had said to at the very beginning was, instead of being, you know, rigid clinician, this is my way, this is my view, she was very sort of open and engaged and said, I want to find out what I didn't know. I want to be challenged. And so you want clinicians who are in that sort of mindset. So go Katrina. Yes. As part of your team. And I mean, ultimately, that's a characteristic you want for everybody who's part of the collaboration, is that openness, that dialogue, that willingness to engage, and that willingness to say, I have no idea what you're talking about, or you're just wrong, or, you know, this isn't right, and, you know, and having that authority, that, and that sort of ability to do that and feeling open and respected and heard. And when you're not, even when your opinion isn't taken into account, you understand why. So with the attribute development, you can't have that laundry list that we had at the beginning. So things do have to get cut. Everyone was sad about whichever one got cut that they didn't want to get cut. But at least if you have that open and honest dialogue, you know, and sort of that rational basis of saying, you know, we heard you and here's why we couldn't do X. Then at least people, you know, you're heard, you're making sense, and that there is really a clear and true process, you know, behind that. Some of the things that really also came up that are probably more challenges that we didn't have solutions for are, how do you think about the demographics in terms of, you know, underserved populations, communities, people who are, you know, have multiple jobs a day, they're not going to have the time to commit, or whatever time they do have is going to be very limited, but they do want to engage in some way. So how do we reach out to those patients? How do we get there? How do we hear their voice? You know, what can we do about that? We don't have a solution for that. I don't think anyone does, but it's something certainly we want to think about as we move forward, and something, you know, Brett talked about earlier about in terms of demographics, and just understanding that that's a study limitation, not just for patient preference studies, but other types of clinical research. And one other interesting factor, I think, that made this collaboration a natural collaboration, but maybe difficult in other areas, is when you're talking about chronic disease versus an acute disease. So if you're talking about a condition where you go in, you get treatment, and you're ultimately cured, that person is a patient for that acute period of time while they go in and get their procedure or their device or what have you, and then they go back, they're feeling good, they're no longer a patient, so they may not have that same interest in being engaged, or there's not really a natural patient group. So what can we do? Or are there ideas or thoughts to how to engage those types of patients who may not consider themselves patients anymore, but their perspective would be helpful to help inform new, you know, medical product development. And ultimately, sort of one thing that came up, and certainly what we'll be doing as part of this project, will be sort of coming out with kind of our process very, you know, concretely for people to understand what we did, to see if there are certain pieces are going to work for them. Not everything's going to work, but at least outlining and providing that information so that people can use that for their for future studies in different disease areas. So that was sort of our discussion in a nutshell. So I'll turn it over to Brett to go next.
Okay. Well, yours was more important because Barry chose your group instead of mine. So everything in our breakout session, we went through what the survey looked like, what the analysis looked like, and some top-level results. Everything, I think, in our session led to the fact that we found heterogeneity matters. Patient characteristics matter. So we're not kidding when we say, if you've met one person with Parkinson's disease, you've met one person with Parkinson's disease. So what we showed in our session was a number of different ways to essentially slice and dice the data based on what we know about the individuals. And we're fortunate in the sense that we used a method where we can do that to a fairly granular level. The difficulty was, despite the fact that we had 2,740 people, we didn't stratify, which was never our intent, by some of these characteristics that we have since learned mattered. So when we started slicing and dicing down to smaller subpopulations, we had some areas where we just didn't have enough people in that very, very tiny specified subgroup to answer those questions. I don't think it's a limitation of our study, but that's one of those areas where if we want to continue to do this, we might want to target some of those things. But heterogeneity does matter. We find that risk tolerance varies for different benefits and for the different risks, matters differently to different patients, and patients with characteristics that we can observe and account for in our analysis. And I was sharing with some people after we were done. I remember, when you showed us your initial cut at the model, you were just looking at DBS and age, I think, and there was just this one corner that kind of showed up as having higher levels of risk tolerance. And since then, you've gotten a little more granular in your subgroups, and that's where we start to see all of these great results. And I think, so what I learned out of just talking with people about these results at this point and being part of today is that if we had looked at the average person in our sample, meaning the mean of the distribution that we see in the sample, we're going to be missing a lot. And we could come up with the wrong answers. We really do need to look at the distribution of preferences across the sample that we can explain by differences in clinical experience and clinical characteristics, because we find very different things when we start getting closer to those individual patient levels. So this is one of those cases where heterogeneity really matters. And we will see it in our results in the preference study, and I think then they're borne out again in the implications that we see in Sho's model. And then comes the difficulty of, the model predict or can describe for which group certain p-values and sample sizes would be appropriate. And then it goes back, I was talking with someone earlier today, one of the other consequences of heterogeneity, it then goes back to the trial designers and who's in the trial, because which of those groups are you going to have? And if you're trying to do all of them, you've got heterogeneity within those groups, so what does that mean? So that's kind of beyond our remit for today, but that's my next line of thinking, I guess, based on what I've learned from today.
Great, thanks, Brett. So we had a really fascinating and fruitful discussion in our AIM-3 session, and it touched on a lot of topics, including things from whose preferences get reflected into our model. How do we broaden the framework to not just think about this in terms of p-values, but really as a guide for decision-making and a tool for decision-making. Also, we touched on aspects of things like what would a partial approval look like, and what are the merits of kind of those types of approaches. And we also talked about actually designing the survey and how can framing effects actually affect kind of the results of the model. And so I kind of want to just conclude with a couple interesting points. The first is there's a term in computer science that we use, and it's garbage in, garbage out. And nowhere is that more true than in this model. And so really the work, we're AIM-3 for a reason, because there had to be a lot of work to make sure everything was done properly in AIMS 1 and 2. So we actually have something that reflects patient preferences when we put it into the model. And so I think that's kind of one point that I think is really important to keep in mind as we move forward. The second thing is I want to bring up a point that Murray brought up during this discussion, which is that there's actually not only a difference in the severity between making a type 1 error and a type 2 error, at least from the patient perspective, but there's actually a philosophical and emotional difference between type 1 and type 2 error. And the fact is, when we make a type 1 error, we actually catch that error eventually, you know, as these products get approved, and then they might be taken off the market for whatever reasons, if they aren't actually safe, if it's proven that way. And so type 1 errors are very visible. And so they can have an emotional impact on the people who might be responsible for making those types of mistakes. But on the other hand, type 2 errors, they're largely invisible. If you, and just a reminder, type 2 errors, when you you falsely reject an effective therapy. And so if we make that mistake, it's much harder to catch that we actually made that mistake. And so it can actually be easier, in a sense, from an emotional perspective, not to make that mistake. But the fact is, there is substantial harm that can be done due to the missed opportunity of receiving something that could have been effective, an effective therapy. And so if anything, at least this model at least brings type 2 error into the discussion, where we can actually analyze it in kind of a rational way. So those are kind of the last points that I'd like to kind of make.
Are we turning over to the next panel?
Yeah. So
all right.
We have our closing panel, and I'll invite our panelists.
I don't know who's still up here without me.
Nice job.
We made it. We survived.
Thank you. We might be a little cozy up here, but that's okay.
Brett, I think you're still supposed to be up here.
Yeah, Brett, you're still up here. Andrew, you're up here too. You are on this panel, just in case anyone asks a really hard question about patient preference studies or the future of patient preference. So as my panel gets settled here, and we can pass along. So thank you all for being so active and participating in our conversations. The intent of this closing panel is to really kind of unpack what all of this means. So as I said at the beginning, it's not enough for us to create something really cool. We want to have a discussion about what this means for how we do clinical trials going forward. So I want to start, we have a couple of new faces up here who you haven't met already. So I'd like to start by having those folks introduce themselves, and then we'll open it to questions. I have a lot of questions, but I know you all may have some questions as well. So we'll both get to some of mine and to some of yours. So Greg, why don't I have you start by introducing yourself?
My name is Greg Molnar. I'm a professor of neurology at the University of Minnesota. I've been involved in medical device neuromodulation for over 20 years. Originally trained in Canada, then so had the academic patient experience, then got whisked off to industry to lead all of the research for neuromodulation for Medtronic. I was there about 10.5 years. Many interactions with the FDA and other regulatory agencies around the world. A lot of inventing and development and research in the space, and then looking for something different, did a little bit of work with the MDIC and proudly became their very first fellow with Susan Alpert, which was great recognition, and then got recruited to the University of Minnesota, so very much continue to be active in this space. In fact, we're working on a Corey grant together with MDIC and others, so really active in this regulatory science space.
Great, thanks Greg. Owen, can you introduce yourself?
Sure. My name is Owen Faris. I direct the clinical trials program in CDRH. I've been with the agency about 15 years now in various roles, but have had a lot of involvement in clinical trials through all of that time, and have been involved with MDIC since its inception. I've had sort of peripheral involvement in this particular project, but a lot of interest and involvement in many issues that deal with incorporating patient perspectives into clinical trials.
Great. And Lauren?
Hi everyone, Lauren McLaughlin at the Michael J. Fox Foundation, and I have the great honor of working with the lovely ladies on my sides here. I primarily work with our entire patient community, our patient council. Brett mentioned that I worked with the volunteers specifically for this project to do some of the pre-survey studies, and then I was also the project manager for Fox Insight, so the online study that this survey was deployed within.
Great, thanks Lauren. So Owen, I maybe want to start things off with a question for you. Since you've been peripherally involved, you've known what we've been up to over the last year and a half. But given your vantage point at CDRH, what does kind of the work we've done here and the work we've presented here today, what does that mean for you as in your role at CDRH?
I think I can go a lot of different ways with that question. So, I think that this is really an extension of a lot of activities that have been moving in this space more broadly. So, last fall, FDA had its first patient engagement advisory committee meeting public meeting to discuss this, the broader issue of how do we bring patients more into the design, particularly of clinical trials. And, I see this as an example that sort of embodies a lot of the discussion that was there. One of the elements is around trial endpoints, and we've been talking a lot about that today, but there are a lot of other issues for which patient input is really sorely missing in a lot of clinical elements, in particular like designing trials that are practical, where patients can reasonably participate and want to participate, and it works in their lives. So there are many different issues that we can think about. But I think that this is, sort of the next extension. And when I think, a lot of questions and conversation today came up about, sort of how FDA staff might embrace this and, the challenges or questions that might be there. And so I've been thinking a lot about that today. And, I would just emphasize that, the FDA staff are always wrestling with this question of benefits versus risks and what is the right decision to make for the patient. I can't tell you how many hundreds of meetings I have been in where we're in a conference room and we have a set of, results in front of us that are ambiguous, right? There are good things that happen, there are bad things that happened, and there's a whole set of unknown unknowns, right? Because we have imperfect data, and we try to wrestle with how do we make the right decision for the patients. This really is, and I think many FDA staff, appropriately so, see themselves as one of the protectors of patients who might otherwise get a bad device. And at the same time, we wrestle with, the discussion that's been out today that there is risk of not bringing a device to market as well. And it's a much less discernible risk, but it's still an important one. And so we wrestle with this every day, and what we don't have is good data in many cases. And the vast majority of those discussions that I've been in over the years don't have reliable, informative patient information. We have statistics of the adverse events and the positive outcomes that we saw, but we don't have patient perspective to help us inform. And so we use a lot of our own thoughts and feelings about how do these benefits match with these risks. And I'll say, that, we bring a lot of passion to that. Many of us are patients ourselves. I'm one myself. I've had type 1 diabetes for 30 years, and I bring that experience into how I help make decisions and inform those decisions. But we do see ourselves as our job is to be critical and to ask questions and to also bring technologies to patients. And so, to me, what's exciting with this is that we have this sort of amorphous question all the time. And here's an example of how we could get somewhat objective answers to that question. And I'm sure that we would have more questions about it the next time a submission comes in that is somewhat based on this kind of data. But the idea that we could develop a tool and we could validate it to get a much better perspective on what the patient really wants, I think is something that we would all welcome. That's not to say that it would be easy the first time it comes in. So, anytime we try something new, there's a learning curve associated with that, and there's a certain comfort level, and we ask a lot of questions. And that shouldn't imply that we don't want to see this be successful or be part of what we do every day, in the years to come. But, anytime we're talking about something new, whether it's, early feasibility studies or breakthrough devices or real-world evidence or bringing patient perspective into clinical trial design, my expectation is that the first few examples that come in and support a regulatory submission, we're going to have a lot of questions, and there's going to be learning on both sides. And what I would say there is that please come talk to us, please bring those challenging questions. And as you do so, you're not only hoping to have a pathway for that device that we're talking about in front of us, but you're also contributing to the whole ecosystem of learning. And we get a little bit better the next time because of that hard conversation we had the previous time.
Thanks, Owen. Greg, I'm wondering if I might turn to you here. Just given that you've done a lot of work in clinical trials in the Parkinson space historically, what were some of the key things that you've kind of learned here, and what are kind of some of your thoughts about how we might proceed with this body of research?
I think I mentioned about understanding the patient and what they want and their suffering is very important. One thing that never gets old is seeing the benefits of DBS. Excuse me for a sec. And that how could we deny that to a patient when we can see that, you know, so it never gets old is seeing that patient in their worst symptoms and turning them on and saying, my golly, look at, they got their life back. And so when you think about having the patient in that model is that if the patient is truly, you know, suffering with their symptoms and we can offer them something that will make such a difference, we have to understand that relative risk. And I think what's also important as I, you know, work with the MDIC and others is demystifying the technology. So today, a textbook surgery would be like what Michael J. Fox had in the late 90s, a thalamotomy or subthalamotomy. Today, someone might do where you're doing all the same surgical risks. So 99% of the risk would occur by the textbook. You put a probe in the brain and you would irreversibly burn, crudely burn tissue in the brain to relieve symptoms. Where today, all that risk, I think, in the medical device is done by surgical standards. So the only risk that's being added is those impacts of the electrical stimulation and things. And so when we demystify that, it's also important for us to have that. And then also understand for the patient that if they would like something to be done, and the part that's actually the risk from the, you know, from the FDA's concern and the technology is probably minimal to what the initial perception is. I think that's very important too. So to listen to the patient with what they want, but also understand the technology and what it means and be comfortable with it. So I think that's very important. I think, and Jeff, if he's still here, you mentioned earlier about the reviewers being comfortable and aware of the patient tolerance and the risk, because they're the frontline reviewers and they are seeing a lot of different topics, a lot of different things that they have to know and are, you know, overworked and to make those decisions. I think they would be comforted in knowing what the risk tolerances are so that it isn't such a the perception of kind of a blocking process. It's one that facilitates to say, here, there's great safe technology, this is what the patient wants, it makes a huge difference, we can all feel comfortable about getting that out the door. So I think it's really demystifying the risk, demystifying the technology, working together, and it will help these innovations get to the patient, because that's sort of the objective at the end of the day, isn't it? So, yeah.
So I'll open this question to any of you who want to respond. We've talked today about how what we've put forward here is something quite transformative. We like to, I mean, maybe it shouldn't be, maybe we should have been taking patient considerations into account the whole time, but it wasn't always the case. So now we've done this, we've demonstrated a model. Now we have to think about shifting the culture. And what can the groups represented here, the groups you all represent, do to kind of start to shift that tide? Or what do you think the task should be that we should do to start to shift that culture?
I'm happy to chime in here. I think the more we talk about the importance of it, the more it will be likely just to become commonplace. If we can disseminate this information, if we can publish on it, if we can talk about it, if we can show that it can work, hopefully others will follow in the path. Just to kind of demystify that it is possible to engage with patients in a meaningful way, in a quantifiable way, that we can then use the data to inform all of our decisions. Hopefully this will become commonplace.
I've got a couple neat examples, just quick. One time I was in a review, I won't say with what regulatory agency, but we were talking about the stimulation, and one of the comments were, you know, what would the frequency be? And we said, oh, you know, 135 hertz, maybe 150 hertz, you know, pulses per. And one of the reviewers went, ooh, that sounds painful. And so this very visceral reaction to something that it's not that you're strapped to the hood of a car at 135 miles an hour. It's a visceral reaction to that, you know, kind of not understanding what frequencies and pulse widths and amplitudes and materials are. And as the conversation ensued, it really wasn't about demystifying it, because even though you could say, well, the charge densities, this and the parameter, it's all safe, just that visceral reaction was really kind of dominated the consideration and the review. And I find that the time we spent this on the company side was to really help people feel comfortable with these electrical parameters, and, you know, particularly for neurostim devices, and understand that, you know, how they're safe and how that works. And then, you know, and then bringing a relative risk where, as I mentioned earlier, that you could have that stimulation, or you could have, if that patient could not get beyond a certain parameter because they say, oh, 135 sounds high, well, we can't go any higher. Well, then you would have to either take that patient after they've had 99% of the risk of the surgery, go to another jurisdiction to get it, or take out the hardware and put new hardware in to burn an irreversible lesion. And so to kind of put everything in perspective, it's all about relative risk. And as soon as that argument came forward, and they said, would you rather knowingly have an irreversible lesion of many, 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 many neurons, millions of neurons, or understand the science and the theoretical risk of maybe a couple of neurons in the brain, in the case of DBS as being a higher, you know, class 3 higher risk. And then it all came clear, and they're like, yeah, we sort of get it now. And then, of course, now we're talking about the patients. It's like, I need this, help me out. I'm either going to do this, or I'd like to do this. Which one, you know, can I have? So.
To also tag team on that, the visceral reaction of living with a disease, I think is something that we also always have to keep in mind at the end of the day, that it's for the patients. Yeah. They are the end user, they're the customers, they are the ones that we are all here for. So.
Yeah, I mean, I think both of the previous two comments are pointing to the idea that we need to keep building upon the work that we've previously been accomplishing. And so, you know, where, you know, a year or two ago it was, we need to be incorporating the patient voice, but we, you know, maybe don't have the right tools to do that. And now we have an example tool, right? And so now I think the next step is, you know, I sort of keep thinking of Jerry Maguire and show me the money, you know, like, you know, well, let's build upon it and use it, right? Like, let's see an example where we can use it. And again, I'll go back to, it won't be an easy conversation those first couple of times. You'll get plenty of questions that will need, you know, some prolonged discussion to address, but that's the next step, right? And then I think the next step after that is, you know, how do you think about a tool like this in other spaces, you know, other disease spaces where there are similar kinds of active patient organizations where we could, you know, build a similar tool, and again, you know, maybe build on these methods such that we can answer real questions that bring a new technology to patients.
I liked Greg's use of the word demystify, because I think a lot of what we're doing here is doing exactly that. We can talk about patient preferences, we can talk about designing trials to reflect patient preferences, but it's all abstract. And I think that CDRH and MDIC have done a great service to the extent that we're demonstrating how these things can happen and what they might look like, you know, warts and all. Every demonstration project is just that. It's a single demonstration project, but you begin to demystify it. It's no longer completely abstract. And, you know, from the, you can imagine, and I don't work for a sponsor company, but, you know, you can imagine that no sponsor company is going to want to take the risk. So it also de-risks that by saying, hey, here's something that we've kind of test-driven that might work. And then to Owen's point, you know, we've got some experience, we still need more, but we've got a starting point. That's how the culture changes, is by those things actually happening. That's the show me the money thing. And I think that's exactly what's happening here. And we've seen it already. We saw it with, you know, with the obesity study and CDRH's use of those data. All, you know, people start talking about that and start thinking about along those lines, and I think the same can happen here. So it's about demystifying, de-risking, and I think that's exactly what we've accomplished, if nothing else.
So we've talked about kind of getting to that show me the money project. What should MDIC and CDRH be doing to kind of encourage the next steps, whatever those next steps may be? I'm looking for some free ideas on what we should do next.
Sounds like the topic of the next meeting.
So maybe I'll redirect this to Greg. Maybe, Greg, you want to tell us a little bit about our project, our next project that we're working on?
Yeah, so the PCORI, so we thought that would be a great area to tap into. And for my department, we hadn't done one of those previously, and it was interesting. And so Stephanie and I and Heather and the gang kind of, pairing up to really talk about neuromodulation again. And we broaden it to pain or DBS, where there are so many different parameters for the device, and most of them have just been used in a narrow range for probably all the same reasons. We know about, one frequency and a pair of, pulse widths, but the idea that in theory, each exploration of a new duty cycle or pulse pattern and stuff would require, by the traditional sense, a whole separate trial. Where here we could say, we know at the end of the day where the rubber hits the road, the energies are safe in the brain, and if we could give the patient, the user of the device, the ability, by their preference, to tune the device to themselves, a tailored medicine approach, and have that flexibility with the understanding, we document, but the fact that it's all safe, but the pattern is different. And it reminded me kind of as we're creating this, talking with Victor before he retired, about DBS and closed loop. And today, the default mode, and again, just demystify this a little while, 24/7 stimulation, basic mode of stimulation. When we talk about something like closed loop or on demand, only when you need it, well, the duty cycles that are revealing themselves are like 40% on, 60% off. So if you were worried about tissue damage, if that was the worry, and if it were legitimate, which it's really not, then it would only be better. And so the idea that we would want to be motivated to have people test this, because less energy means a smaller device, means all sorts of benefits to the therapy. So the idea of vetting all these possibilities quickly is going to be important. And myself, having been at Medtronic for years, the original DBS system, and then the Abbott system came out. And it's interesting because it's a new device, but by a new device, someone very close to this equipment, the quality, the way it's structured physically, any interaction with the body is actually better with the new technology, and some of the features of the new lead to be able to steer the field, and I've worked with that for years. And so these are all things we should be able to really test and have patients test for themselves, and that could prune the tree a lot faster. And so we'd learn a lot faster, understanding that basic premise is the energy is safe, and then we can explore. So that's a big part of our study, is to gather those preferences for testing new modes, closed loop, field steering, and the like, but across the different types of therapies. Pain, which obviously was a topic here today too, and PD, but then for the deep brain stimulation.
I think that gets to just sort of a broader point about the idea that whenever we're thinking about a clinical trial, we have to think about it in the context of what we potentially already know about a particular technology. And so we don't need to reinvent the wheel every time, but we need to think about, well, what are the new questions? What are the potential risks? And how do we design a trial that's fit for that purpose to answer those questions? We no longer, need, if there's a new defibrillator that comes out, we no longer need to demonstrate that defibrillation is good for people who need defibrillation. We need to show that this one works, right? And it does what it's supposed to do relative to what we know in that space. And I think, that concept applies broadly and across many disease states.
Yeah, definitely.
I have a question for actually Dr.
Okay, you can ask a question.
So you were talking about what's next for kind of MDIC and maybe CDRH, but I was wondering, since we started with Andrew in the morning, and Sho has done such a great job on this project, what's next in this area for you guys?
So I was hoping to avoid saying anything on this panel of experts, because it's better for people to think you're dumb than to open your mouth and remove all doubt. So I think from our perspective, the limited academic perspective, we think that there are two different next steps that we'd like to take. One is on the analytical side, to try to combine the different kinds of measures of patient preferences in a more rational way, so as to reflect overall outcomes that I think all of us are looking to achieve. So rather than using one dimension to combine multiple characteristics in a rational fashion, that's sort of what's been happening here in this MDIC, Michael J. Fox Foundation, FDA collaboration with Brett and RTI. We're looking at different characteristics, but ultimately trying to boil it down to a measure that we can communicate. And I think that's the second challenge that we're focusing on, which is communication and narrative. I think that people fix on p-values because we know it and we're comfortable with it. At least we think we know it. But the fact is that in order for us to replace p-value with something more useful, we need to provide that alternative. It takes a measure to beat a measure. And so we need to come up with something. And at the break, we were talking about if all of us were Bayesians, that would be a lot easier because we can simply use posterior probabilities in order to make our decisions. I got to tell you that for most people, the term posterior probability does not inspire confidence. It inspires something else. But, so frankly, I think coming up with better narrative for patients, for doctors, for scientists, for other stakeholders, that they can hang their hat on and say, yeah, you know, this is actually a better approach than focusing just on p-values. So it's a combination of better analytical tools, but then better communication to be able to take those tools and allow other stakeholders to be able to embrace them.
So I'll point to the end of the table then, because you guys tell the Fox Foundation what they should do all the time. What do you think we should do next? We, MDIC, CDRH, this is your chance to give us some, tell us what we should do.
Don't be shy now.
Clearly, what has happened here is unique. This situation was ripe for this work. The right players came to the table. Fox Insight existed. 18,000 people were ready to fill out questionnaires. It's a very unusual situation. And yet what you've done, I think, has a lot about it that's probably replicable. So my thought is get the word out there. Go to every conference, publish in every newspaper, magazine, and newsletter. Let people know what was accomplished and can be accomplished.
Yes, I was going to say the same thing. Rev up your marketing teams. I mean, as I said this morning, if we have this nice workshop and then nothing happens, we have failed. Mm-hmm. So we need to broadcast it and show that it can be done, and show that it can be done in different contexts, because it's certainly not something that is unique to Parkinson's. In fact, Parkinson's may have been quite different because of the heterogeneity that you described. Some other, cancers or other things are much more homogeneous and might be easier to gather data about. And so you need to get the word out because you guys have the platform, as do you. And I can go, tell people till I'm blue in the face, it's not going to have the impact. And the only other thing I would say is that whoever it was who said we need to incorporate the patient perspective into other aspects of this, no patient would ever name it deep brain stimulation. I mean, the only thing I can think of that would be worse would be deep brain drilling. I mean, it's the worst name on earth if you're trying to sell this thing. I mean, it's scary. It's a horrifying name. It may be accurate, but it is not helpful. And that's just an example of, somebody came up with this name that didn't consult a single patient, I'm sure.
Right. Well, in academia and in government, we are all about the acronyms. So, we maybe need to find a new acronym for, because it's an important point, and it's not one to be taken lightly. And I think we even saw this, Brett, on some of the interviews with the patients that we did leading up to, before we launched the preference surveys, that there were a number of patients that already had very deep-seated ideas about devices or about stimulation, and some of those may just come from, frankly, biases because it sounds scary. I want to make sure we have a chance for all of you to ask questions. Pick on whoever you'd like. Ask whatever you'd like of whoever. Annie, I don't know if you get to ask questions. Oh, you're going to walk around. Okay. Right up here, in front.
So thank you for this one. Oh, hold on.
Oh, she's coming with the
Annie's
microphone.
coming.
I can't run that fast. I'm wearing heels.
So thank you for this wonderful day. It's been very, very informative and I think very important. I'm Sue Manoff. I'm from Merck. And my question is for Owen Faris. So we started out today with a wonderful presentation from Dr. Lowe about different p-values for different levels of types of diseases, and you gave the very interesting perspective of glioblastoma as compared with prostate cancer. Is applying different p-values to clinical research from the regulatory perspective, different sample sizes for trials, different requirements for licensure, is that realistic?
Yeah, I think it is. I mean, I think that it's new, and I think it'll challenge us, and I think we'll have to have a lot of conversation about the robustness of the data, and this is an example of how one can get robust data that can support that conversation. But I think that's where the questions will be. We want to make sure, I think, review teams at FDA are appropriately skeptical that one might cherry pick certain data to design a particularly liberal trial. And so I think there'll be a lot of questions about, show me that this is a robust method that really does represent what patients believe and what their priorities are. And those first conversations won't be single meeting conversations, but absolutely we wouldn't be here in this meeting. We wouldn't have gone through what we've gone through thus far if that wasn't part of the discussion for the future.
Is Martin allowed to ask questions?
Maybe.
I have a feeling Martin might not be asking a question.
Thank you. I wanted to ask colleagues from the Michael J. Fox Foundation about if you have the sky's the limit, the ideal state of collaboration between your foundation and CDRH or even FDA. What would you think we can perhaps further our collaborations and perhaps even involving our largest group of stakeholders besides the FDA? So I want to hear from you.
I was hoping that I could defer. I think that this has been a great starting point. I think that just the fact that at the foundation we exist to find a cure, to put new therapies into the hands of patients, but the approach that we have followed is to de-risk along the entire therapeutic pipeline development process. And obviously the FDA is kind of one of those endpoints there, and so we as a foundation have been working a very long time to get there. I think we have tried knocking on the door on a few occasions, but what's so unique about this project is you approached us. And so that bottom up, top down mix and meeting in the middle has just been so wonderful to be able to collaborate on, to bring the various voices, the various experts and stakeholders together is great. I think this was the first iteration of doing that. I think simply looking at the way that we have sliced the data, the questions that we asked was our best guess, and so we can certainly always iterate and improve upon. That's what science is, and so I think it's just the beginning of a beautiful relationship, and it truly is, and that sounds somewhat cliche, but I would like to say, and I know that many people at the FDA say that they are very patient-centric, that they are trying to develop new projects and new programs, but this is a true illustration of how that can be done. The doors are open and you are willing to engage in dialogue. So look forward to continuing that conversation. Anything else?
I guess we're on the FDA circuit now. I'm getting my steps in, so I guess that's a good thing.
Hi, Carlos Pena, Director for the Division of Neuro and Physical Medicine. And it's reassuring to hear the discussion about patient preferences and the outcomes, because on behalf of the staff that is several dozen strong at the agency, I know they take very seriously the many different factors that should be involved when making a decision to approve or not approve something. The technology, the IFU, the patient population, the evidence data, the certainty that we are comfortable with, the weight of the evidence. That's, that pulls into statistical considerations. And so I think one recommendation that I think follows very closely to the patient input that we just heard from the panel, from Margaret and Anna, is that if we can, I'm going to steal a word from you, Owen, if we can find the right fit and make sure the fit is fit for its purpose here, and how we add that to the decision process, that would be a great win, I think, for everybody. So again, if we can continue this discussion, I think you're going to have a lot more staff, at least from my division, listening and looking forward to how this dialogue continues.
All right, we're just continuing the FDA Roadshow.
I'd just like to comment that it was really great to hear Carlos's input on that, because at the end of the day, legally, that's still where the rubber meets the road. And the understanding that, a relatively conservative regulatory body is willing to look at new approaches and to continue to grow and innovate is probably the most important outcome of this. And, as we've heard, Margaret said several times, if, it just kind of dwindles out here and doesn't develop further, we truly have failed. And so, as I asked in the previous panel, how do we empower FDA review staff to make these types of decisions? And I kind of really liked what Andrew said in response to that, or maybe before that, that perhaps we get together, and that may be a part of what Jeff was talking about with the collaborative community, that maybe these very weighty decisions should be shared by all of the stakeholders in some way. Of course, in the end, by law, there will be, the regulatory group that's responsible for the ultimate decisions, but having the sufficient input from the other stakeholders and having this occur on a regular basis, and as we learn and grow more and we innovate and we change, so that we can adapt what we're doing to the changing thinking of society, will be best for healthcare, and I won't even say patients and people.
Woohoo, not FDA. I don't know why I'm giving commentary as I walk, but I am. Just to keep it entertaining at the end of the day.
This is what happens when we give Annie a microphone.
So I was curious about the data you showed on the race and ethnicity, educational attainment, and also thinking about things like age and gender of patients in a patient preference study compared to the population, or at least the population with the disease state as a whole. I'm curious what others thought of those numbers and what you would think of that in designing a study.
So we have a couple approaches. So let me step back. So when we have a sample that is kind of skewed, like we have here, doesn't invalidate the sample, it just limits our generalizability. So what was unique about this, and we're actually finding a lot now more often when we work with patient organizations, is we can sort of pre-specify the sample and stratify and set it up more along the lines of what you would do, have stratification plan and a sample frame
that gets us exactly what we're looking for. In this case, when we worked with the Fox Foundation and Fox Insight, we knew what Fox Insight looked like, at least at the time, and that was evolving as we were going along. And so we knew that what we would get was probably representative of Fox Insight, but maybe not representative of the larger population with Parkinson's disease. But that we didn't let that stop us, essentially. And when you do patient preference studies, the problem that we have in this particular case is when we have true underrepresentation. So here we are missing some key parts where we would need to oversample. But if instead we had kind of the proportions that were wrong, but we had sufficient people in each sample to be able to look at what that group's preferences were relative to the other groups, that's okay. Diversity is good. Actually, diversity is probably our watchword that's more important. So I'm less concerned about representativeness than I am about diversity. But then again, in this case, we're lacking some diversity. If we were actually informing the development of a specific trial for a specific product, that would have been critical. Because this was proof of concept, because we were working, I don't want to diminish this, but we were working with what we had. You know, we had access to this great trove of patients. We weren't going to turn that down simply because we weren't going to get every different type of variation in diversity under the sun. But if we were to work with somebody to do a patient preference study, to design a trial that would go to FDA for that type of discussion and review that Owen was talking about, we would need to start at the beginning and say, all right, who's going to be in this trial? Who's eligible for this? What does your indication look like? And make sure that that is representative. So we had the luxury here of being hypothetical. In the real world, we would need to be much more attuned. But I just want to go back quickly to the issue of working with patient groups. Working with patient groups is a wonderful experience and probably a very good way to go. But the patient groups themselves are not necessarily, in terms of characteristics that represent diversity, representative of the patient population as a whole. So there's a trade-off there, that with having a great partner to work with who may not have access to every type of patient that you're looking for. And that's a consideration we run into all the time. And for sponsors, that's something, and for FDA, that's something that we'll need to think about how to handle that.
So unfortunately, the FDA track is back. So I have a question actually for you all at the Michael J. Fox Foundation. In light of what was just said and the movement towards trying to include patient-generated data in decision-making by FDA, are patient organizations now getting more activated to include more diverse populations with respect to the spectrum of disease, with respect to demographics, other things like that, so that it can be informative and generalizable to inform a FDA decision?
Yeah, certainly. So I obviously only speak from the Fox Foundation perspective, but that's a major initiative that we have. Not to steal words that have already been used, because we've been using it as well. We aim to demystify what it means to participate in clinical research. So a lot of it is just education-based, of understanding not only the disease, but then also what it means to take action to move towards a cure. So there's a number of different mechanisms that we're doing in order to try to educate the community and prime them in order to participate in research. Just to define what it means, to understand the different types of trials that might exist, that it's not all being a guinea pig, that there's a difference between interventional and observational studies and things like that. And hopefully once we can define a vocabulary that resonates, then also taking those to underserved populations. And so there's a number of things that, again, at the Fox Foundation, we're doing pilot studies, working with influencers in certain communities in order to understand what might resonate with those communities. How do we educate and involve them so that it truly can be more representative? It's all a work in progress, but we're very excited to be able to do that in order to find ways to bring the voices of many patients to venues and platforms like this.
If I could just add to that, I've had the great fortune of working with a number of patient groups. And, you know, this is a struggle for many patient groups, is how to really work collaboratively with underserved communities where that kind of access to research has always been limited, that kind of engagement has always been limited. And that requires a cultural shift too. And we're seeing a lot of cultural shifts from the patient perspective, where patients are empowered and patients are eager to learn how to become engaged, like Lauren was suggesting. But there are communities where, based on lots of different characteristics, where that requires extra engagement, and that requires resources and time. And there are certain disease areas that are well funded and can do that, and then there are others that are not. So, you know, I think everybody agrees that that's a laudable goal, but that may take time, particularly in certain disease areas where you don't have well-funded patient advocacy organizations. That may be slower. That's only my opinion based on limited data, but that's what I've seen working with different patient groups. That's a big deal and a big challenge, and it's just going to take time.
Yeah, to just add on that as well, one of the reasons that the foundation decided to develop the Fox Insight study in general was we were hearing from so many of our community members that they were willing to participate in research. They wanted to share their voices. They wanted to share more. And as Brett was saying, there's kind of that cultural shift that is taking place slowly but surely, where I think as patients have better access to information, they are more demanding of getting even more information, of driving things even faster. And they're starting to understand that they need to be participants in that. And so finding ways to collect that information in a systemized manner, such as Fox Insight, is hopefully just one way of doing it. It's one way that we're trying at the Fox Foundation.
Can I make a comment? Because there's something that's been brought up by the last several questions and responses that's really important, but I don't think we've actually hit upon it directly. And that has to do with patient stratification. So I think we all know that personalized medicine is a reality today. And that the idea of being able to stratify patient populations based upon various different characteristics, that's actually a very powerful way of, first of all, finding a particular population for which a therapy can be extremely effective, but it's also a way for bringing more resources into this particular space, because clearly the Orphan Drug Act has provided incentives for focusing on targeted populations, and that's something that can be used in this very case. So the question about whether or not the particular sample that we had from the survey was biased, as Brett pointed out, it may not be representative of the entire population, but nonetheless, this could be a useful subpopulation in its own right. That actually highlights the ability for the Michael J. Fox Foundation to provide enormous amounts of information, both electronic medical records as well as genetic data, to allow device and therapeutics developers to stratify the patient population to create subpopulations for which these therapies can be extremely effective. So that's something that I think we haven't really discussed, but that the use of subpopulations and the use of the heterogeneity of Parkinson's patients to the advantage of developing these customized therapies, that's something that could actually have a tremendous impact. And all you need is to start working on a subpopulation for which you can find a really effective therapy to have that kind of reinvigoration of R&D efforts. A case in point is cystic fibrosis, where the Cystic Fibrosis Foundation brought to bear enormous amounts of information on their particular patient population, and that greatly accelerated the development of a very successful set of drugs.
Fox Insight data will be de-identified, made publicly available later this year. So.
Well, I think that goes, I think in many areas for medical device development, and this was brought up earlier, we may start with a certain population where the benefit-risk equation is a little bit different. And so the acceptance of uncertainty might be a little bit different. I think the example of cardiovascular resynchronization was brought up earlier, where that technology, when it was an unknown technology, was first brought to market for very sick patients for whom they didn't have good options. And this was provided a sense of hope and an opportunity. And as we got more experience with that device, and as practitioners and patients who were a little bit less sick started to use that device, and as trials were conducted in less sick patient populations, we started to understand better how that technology was good for some patients and not as good for other patients that looked a little bit different than those high-risk patients that we started with. And a lot of devices develop that way, and there's no reason why we have to say one bucket, for one certain kind of disease or condition.
Hi, Jem Munger with PwC. I think this is, I guess, more for Owen, but to anyone that wants to answer it. I was speaking with Annie at a break, and she mentioned that there's collaboration across the other medical product centers on similar sort of initiatives with patient-generated data or patient preference information being incorporated into clinical trials. To what extent can you elaborate on that a little bit, first of all, and second of all, to what extent are you collaborating with folks like Dr. Lowe brought up precision medicine, so NIH and their PMI or All of Us initiative, and folks like HRSA for underserved populations through federal qualified health centers?
Yeah, I actually think some others in the room may actually be able to answer some of that better than myself. Maybe I'll just take a stab, but I know Katie and Michelle may have some other thoughts as well. There's a lot of initiatives, this being one of them, where we see a lot of synchronicity and need to move in a collaborative way across the agency, certainly, and with NIH as well. So, bringing in patient perspective is certainly one of them. And, you see not just at the center level, but at the agency level, the idea that we need to do this better. We haven't done it well historically. We need to do it better. Many other initiatives look a lot like that. So again, real-world evidence is another thing. We can get data in a smarter way than we've been doing it historically. Let's work across the agency and across other partners and throughout the federal government to do that. I'm not sure that I can speak as well to the specific activities, but Katie, do you want to speak to that at all?
And I'll start walking and then I'll say something and hand it over to Katie. So we are collaborating with our sister centers, both in Cedar and Seaver, in terms of a lot of the patient science activities as much as we can. Of course, we have certain statutory limitations that affect some of the things we can do, but that is definitely an ongoing, robust conversation internally.
Yes, across the science of patient input, where we include in that patient preferences as well as patient-reported outcomes, the 3 medical product centers have, as Annie said, ongoing, robust discussions about typically either looking for methodological scientific questions that are challenges oftentimes, and this is true in other areas like in biomarkers or other types of outcome assessments where the science is in a certain place and there are particular gaps that are priorities for regulatory application of that science. And so, the role for FDA in advancing the science is to make that more clear and then to find ways through collaborations and partnerships and other means to close those gaps over time so that we can bring those scientific uses into regulatory use. I think patient preference is a perfect example where there have been, numerous, I think, what did you say, Brett, at the NASA meeting, over 100,000 hits for patient preference if you do a lit search. But, it shrinks down when you look at benefit risk applications, and it shrinks down even further when you look at regulatory use. So, those are the kinds of things that are sort of cross-cutting across the medical product areas that we're looking to collaborate with our sister centers on.
Dean Brunding, CDRH. Part of the important process in the regulatory decision-making for FDA is possibly taking a PMA or a de novo device through the advisory panels. As part of that process, this information that we're talking about is pretty new. It's possible this data could and probably should be part of those panel deliberations. So what would FDA do to prepare the advisory panel to be able to, evaluate and assess that information, which could have a big impact on the assessment of the panel with regard to the decision, yes or no?
It's a great question, Dean. So, we've done this before with other things that are new with advisory panels. So, the first time that we had a PMA rely very, very heavily on computational modeling and the clinical data were really secondary, we spent the first couple of hours of that panel meeting explaining how computational modeling was really a real thing that people could believe in and count on, and and try to distill it down to something that was digestible from by people who have other areas of expertise. There are other things, when we bring trials to a panel, there are many, many panelists who still aren't very comfortable with Bayesian statistics. And so when we're not talking about p-values and we're talking about, different kinds of ways of characterizing data, we have a little tutorial on Bayesian statistics. And, I think that, your question really brings to mind, some thoughts that I had as Dr. Lowe was presenting his presentation this morning, which is the challenge to me, and I think you did it very artfully, of presenting some very complex ideas to a very broad audience that includes people who have been, had PhDs in statistics for years and people for whom this is a really novel concept. And so I think we have to do that. Frankly, we have to do that with our review teams too. This is going to be new to us. Many times when someone brings a new device to us, forget about all of the technical elements we're talking about right now. We have a company comes in and they've been working for years on the technology that they know in and out and very deeply, and they have to get us up to speed in the span of a few weeks or months, and we ask a lot of questions and we have to all get to a comfort level, and this is going to be the same thing. So we will have to have that discussion about how to incorporate these kinds of concepts with the review team, and then the review team and the company will have to have that discussion with a panel, and we'll have to do it in a very methodical and thoughtful way so that we can have the right kind of conversation that we need to have at the end of the day.
Back on the FDA circuit.
Well, I just have a comment that goes actually past FDA. As we're all aware, a regulatory approval or marketing clearance only gets us part way. Payers have to make decisions that they're going to cover and reimburse the products. Physicians need to adopt them. And of course, finally, patients need to accept the use of devices based on different types of evidence than they're otherwise used to. So I would strongly suggest that we keep this in mind, and Owen, as you start to develop the educational process for the panels and the review staff, let's keep in mind that we need to also educate the other stakeholders early and often so that what we do at FDA doesn't fall on deaf ears.
We've answered all the questions. There are no more questions. All right, well, I want to thank my panel both for the work you've done up in this project and for being here today with us to help us figure out what this all means and why this matters. So thank you.
Thank you.
So with that, oh, see, look, he just walked into the room, right? Right timing. I'd like to turn things over to Barry Leiden from Edwards Life Sciences. Barry is the chair of MDIC's Science of Patient Input Committee, and I've asked him to share a little bit about the work we're up to and how this kind of all fits into context. So, Barry?
All right, does everybody have an agenda? Can you see what my role is? Like where I sit on the agenda? It's right before we leave, okay? So I recognize I am standing between you and the weekend, essentially. So I'll be crisp. Thank you so much, MDIC. Stephanie, you did a fantastic job. I think we really enjoyed this. I think we got a lot out of it. And kudos to everybody that was involved in this project, particularly the patients who have donated their time and energy towards this. It's really admirable, and I'm humbled actually to be in your presence. I wish I could say I was a patient scientist, but is that, I don't know if I can say, do I want to say that? So thank you very much for joining us. And also, thank you, FDA, for providing such leadership in this area. If it was not for FDA and their leadership, we would not all be here. So as we opened, Pamela and Stephanie both mentioned that MDIC is a public-private partnership between industry, academics, patient organizations, and government. And we have this group called the Science for Patient Input Steering Committee, or Working Group, which is a fairly large group, and you are welcome to join us if you're interested. And our purpose is really to try to find out ways to develop greater scientific tools to help sponsors, and that could be academics, patient groups, or industry, to have really what we're thinking of as regulatory science, really tools that can be used to help get products to patients, and really from a patient-centric perspective. And the first project that we had was the Patient-Centered Benefit Risk Framework and Compendium of Methods. And if any of you have not had a chance to check that out, you should. It's really extremely good piece of work. In fact, it's being referred to globally by multiple organizations as a reference point to how to look at how to elicit patient preferences in decision-making, not just regulatory decision-making, but really in any decision-making process. And from that, we are embarking on a number of different projects. This is one of them. But some of the other projects that we're working on all are very much focused on the various stages of the total product life cycle. So, but with a lot of emphasis, because we got a lot of feedback from the groups that are part of this project, that there really is, it could be very helpful to get patient input in the clinical trial stage of development. So we actually have 2 or 3 different working groups, depending upon how you look at it, focused on how do you design clinical trials in a way that are more patient-friendly, from a recruiting standpoint to working with patients, and also to how you measure what the outcomes are in terms of what's important to patients and what kind of statistics you're going to be using, like the methods that we were talking about here today. We also have a working group that's focused on communicating those results or the benefit-risk profile to the community and to the patient community. And those projects are ongoing, and we expect, hopefully, with some additional funding in the next few months, that we'll be off and running and have similar types of work product available to you and to the public in the next 12 to 18 months. We also just got approval to start a new approach to this, and that is to actually try to do our own patient preference study using MDIC as the safe place for multiple industry stakeholders to get together and do a multiple industry partnership around congestive heart failure. And so hopefully we'll learn through that process under the umbrella of MDIC on what to do and what not to do, how to do it and how not to do it. And then we'll share that again with the community. So that's the ad for MDIC and the Science for Patient Input. And again, if any of you are interested in getting more involved in any of these work streams, please let Stephanie or me know. We'd really love your participation in any way you can. So with that, am I concluding? I can't remember. I'm it. I adjourned the agenda, so thank you. So thanks everyone for coming, and please get home safely. Hopefully the weather, it doesn't get too bad.